Hello, my name is Isabella Alvarenga, and I'm a PhD candidate with Dr. Greg Aldrich at Kansas State University. Today, I'll be presenting my poster on the effect of resistant starch type 2 on satiety hormones of fasting dogs. Based on previous research, extrusion of dog kibbles with corn at low thermomechanical energy retains some resistant starch and slowly digested starches. Resistant starches are substrate for colonic bacteria to produce butyrate and other short-chain fatty acids, while slowly digested starches release glucose at a slower rate in the small intestine. These may promote satiety by supplying energy to the body long term. Our hypothesis was that increasing levels of resistant starch due to processing at lower thermomechanical energy would result in increased cell signaling for satiety. The study objective was to determine whether or not diets with greater levels of resistant starch would affect satiety in dogs at a fasted state. A single recipe, adult dog food with corn, was extruded at 3 levels of thermomechanical energy to produce a low, medium, and a high resistant starch diet. There was a tendency for greater slowly digested starch in the high resistant starch diet, and the high resistant starch had double resistant starch relative to the low resistant starch diet. Diets were fed to 24 adult beagle dogs for 28-day periods in a Latin square design. Blood was collected at a fasting state at the end of each period, and plasma was separated and frozen until analysis. Satiety hormones were measured in dog plasma by ELISA. These were analyzed as a mixed model using the Glimix procedure from SAS with diet, treatment sequence, period, and carryover as fixed effects, and dog location as a random effect. Significance was noted at a p less than 0.05 and Tukey post-hoc test was applied. Now moving to the results. Satiety hormones, ghrelin, leptin, gastric inhibitory polypeptide, glucagon, pancreatic polypeptide, and peptide YY were not different among treatments. We expected peptide YY to increase because its release is triggered by undigested foods in the colon or rectum, but this did not happen for two possible reasons. First, there wasn't enough resistant starch in the colon to cause an increase, and second, peptide YY is more responsive to dietary fat than carbohydrates. Glucagon like peptide 1 was below the detection limit in 35 samples, so this hormone was not included in the analysis. Surprisingly, insulin was the only hormone that had a tendency to increase in the high relative to low resistant starch diet. The underlying mechanism for this finding needs to be further explored. In conclusion, the experimental diets did not affect plasma hormones associated with satiety at FAST. We would like to thank Hill's Pet Nutrition for financial support of this project. Thank you.